right. Hello, everybody. My name is David Leclerc. I'm the host of the ChUG podcast. I'm incredibly excited today to host Florian Mair and Rui Gardner to have this discussion. I call it the great debates of our time. How should we apply a manual computation on top of our mixing data? Yes or no? Many people have lost their lives over this feud over many years, and we're here today to fix this issue. Incredibly happy to have Florian and Rui. Florian obviously is behind OMIP 102, this 50 color marker. Rui Gardner's group has done they prepared a 13 color backbone panel. So within these two guys, we have a wealth of experience. I have none of that experience, so I'm going to be the voice of unreason, and I'm going to take the position that let's do it, and we'll see where that goes. Thanks again. We're on the back of the CYTO meeting. We came back, and this is a topic that was discussed during a tutorial. And we were discussing before recording. Well, I think I missed it because Anna Belkina was giving a talk at the same time. And whenever Anna says something, I'm going to go see what she has to say. I think Florian, you saw this presentation. Why don't we start with this particular session, and we'll use that as a trampoline to move forward.
Yeah, I mean, I think what was clear from that session at CYTO is that it's a topic that is important to a lot of people. There was great attendance. There were very intense discussions. And I think it's been talked down over the past years for a while, where people say, oh, if you do it right, you never have issues with your data, which I think is not the reality. I mean, you can get very close, but there are sometimes issues with your data, and there are various reasons for that. We can talk about it later. And I think people just want to have some guidance how they can work with the data. And some of the people probably also want to know what is exactly happening, because they just don't understand what's going on. And I think this is why there were so many discussions, where there were so many people. And I guess that's what we are following up on today.
Right. So essentially, there are two people explaining how to apply manual computation after unmixing. Do we have an idea, like a rough idea of why they justified this particular tutorial, or why would they present these ideas? Like, why do they think it's important to do it?
I mean, the way I understood it from that session is that one part of the talk was about a clinical lab that just needs to have kind of procedures in place that allow for like high throughput and getting kind of consistent data, as they called it, out of their pipeline. And the other half of the tutorial was really trying to instruct people how to deal with these adjustments in a more or less systematic fashion. But I guess that's where it starts, because what is systematic, what is not systematic with like an adjustment that you apply to your data. And I think whenever there is an issue with your data, you always need to take a step back and figure out, why has it been happening? Is there a root cause in terms of incorrectly prepared single stain controls? If you feel you did everything right with your single stain controls, was there an issue during acquisition, or was there a sample specific issues? Which is why I'm a big proponent of what I call the reference samples. So for people working in the mouse world, I often see that in my core facility, that people come with like the most complex tissue isolation ever, like the salivary gland or like part of the adipose tissue. And then that's what they stain, and then it looks weird. And then they ask me, is this my panel or is it the tissue? And I go like, I have no idea. Bring me a spleen, then I can tell you whether your panel works well or not. So for the mouse people, it's kind of always have a wild type spleen. And for the human people, what I used to do during my time in Seattle, we just had like one healthy donor, where we had a Luca pack, and like we froze down like 200 aliquots. And every single time I was doing like a staining on like a patient sample or a tissue sample coming in, I would thaw one of these reference donors, because it would give me peace of mind. Because if I know I've stained that donor like 100 times, and if the 101st time it all of a sudden looks different, I know I'm having an issue with my panel or the single stain controls or the unmixing.
Right.
Yeah.
Yeah, I agree completely with what Florian just said. I mean, as scientists, we're supposed to be applying the scientific method, right? Manipulating data or changing the data after the fact based on your assumptions where the data should be or should look like, is not the scientific method, right? And what you're saying, Florian, is I think what I'm understanding is that we should normalize the fact that flow cytometry data is not perfect, and then start building tools, start building understanding of why isn't it perfect, and trying to fix that, right? If you manipulate the data after the fact, I mean, you're just going to hide what's there, right? And you're not going to address the root cause of the problem. The practical reasons for doing it, that's another issue. I mean, we're all humans. I don't think we're the gatekeepers of how to do science properly. I don't think anyone should be dictating, oh, you should be doing this like that, or you should be doing it in a different way. It's more a question of, I mean, are we scientists? Are we following the scientific method? If we are, and Florian just described a few things that you can do, right? Choosing your reference samples, making sure you understand, that there are things that are out of your control that you still, right? We don't understand exactly where all the problems are coming from. They don't come all from the single stain controls, but we know that the majority of problems do come from there, and
Yes,
right,
they
and
do.
we can start addressing those, right? We've been slowly addressing some of those issues, but um, if anything, it actually prevents the community from moving forward, in a proper scientific way, I would say. That's at least my opinion.
I have a comment here about the semantics that is very dear to me because I heard in many discussions that people call it data manipulation, which you did now as well, Rui.
Right.
And just to say something different here, I wouldn't call it like that because we are not changing anything about the raw data. That's the nice thing about a flow cytometry experiment. The raw data, the numbers, they always will be there. So I would rather call it a data adjustment, because that's what we are doing. We kind of adjust matrices, settings, not settings. We are adjusting matrices that will, of course, influence the appearance of the data, but we are not manipulation is a strong word. We are not manipulating anything because the raw data will always be there, and whatever you put on top of it is a correction factor that still keeps the original values. So
Right.
I think that's something that is important to me because manipulation is really a strong word.
I mean, at the end of the day, it is, right? And if you think of the definition of fraud, it doesn't matter if it's on purpose or or by accident, right? That's the thing, right? If you're not following the scientific method, the scientific process, then you risk putting out data, and this is a huge responsibility that we have, right? When we put out data, that's something that concerns me, every time. You always have that slight nervousness, again, putting this out there. I mean, I should be as sure as possible of the data that I'm putting out there, because there's a huge responsibility, right? It can drive people into different directions, and it could take years or decades to get the scientific community steering in the right direction. So there's a huge responsibility there. That's why, I call it, and I'm not being loose about it. When I call it data manipulation, is just to also put that seed in people's minds that, wait a minute, if you're adjusting things towards what you expect, yeah, you're risking changing the interpretation or something like that, right? And we see that it's a slippery slope that we go into. But I do agree that most of the times, or I wouldn't say most of the times because we don't even know, right? But many times, some of these adjustments, right, are harmless. They don't do, but then I would argue, then why risk, right? Why go into that temptation of making your data look good? And I'm sure there, Florian, you can also discuss, instances where it is important to have that data, let's say, looking good, for transformations, for right, for dimensionality reduction, these kind of tools that require the data to be as good as possible, right, as best as possible. So I see the temptation there, but my my advice at least is it's very difficult to teach best practices and do things in a proper way if you're encouraging people to, to change the data after the fact based on your assumptions, right? And the other day I had a really nice discussion. We were during the annual flow course. It was actually in the context of sorting, and somebody was saying that they were talking about, can we sometimes we need to change the data, and we had, it was a negative population and a slightly positive population around
the famous bump, the famous shoulder.
Right. And they were saying that, oh yeah, see, like I know sometimes I need to because I want to sort, and that bump is too close, and I want to get it a bit further away.
Well, that won't help.
Right. I was like, oh my God, this is not the way to go. Actually, that population might be negative, and it might not be totally well unmixed or compensated. And so here we are, we're normalizing this, that it's okay. And I think this is very dangerous. I mean, it's very hard to have reproducible data. How much did you correct? Sure, we can report how much we corrected, but we know that the sources of error and the differences between the instruments also play a lot, right? So it masks all these sources of errors, and it makes it harder to interpret and reproduce. So
but there's a range, right, of corrections that can be applied that will change the perception of how, if you look at data, like what that person is trying to do. Like if you have a 50% correction that you need to apply after unmixing and you do it, then obviously there's a big problem. If the correction is like a few points and you're just doing that for aesthetic issues and
yeah,
then
what do you guys think of for instance, rather than going in the direction of changing, right, the data after the fact, more of finding metrics that can tell us, whether to accept this data, like this data, best practices were used, single snake controls were as best as possible. You get data as close as possible to what you expect, and accept it based on a metric, right? And if not, then we have to go back and really find out what the issues are. Because I don't know if we've discussed this, and I don't know what your opinions are. If we change 5%, if we're changing 5% in a very low intensity signal, that's not a big deal, right? The 5% in a high intensity signal is quite a big deal. So where do we set a threshold here?
I think we discussed it on the podcast before, and we had a threshold of percentage that like ill-defined and it's pretty random, but the general idea is that you also have a string of other methods that you use to justify your data, and everything kind of comes into agreement at the end of the day. So even if your flow data is somewhat corrected after the fact, there's a whole bunch of other technologies, techniques that supports your findings. So there you're on a stronger footing. The end point is making sure that whatever you discovered has some kind of biological meaning.
Right.
And so
I mean, you touched on multiple points. I mean, if I may comment here, I think the absolute value doesn't matter that much, except the fact that it tells you your single stain control was more or less off. For me, kind of the way how I go about this is that first, I always come back to this reference control sample, because there, for a wild type mouse, I've been doing immunology for 10 years, if you're in that field, and you know how marker expression patterns are supposed to look like in a spleen of a wild type mouse, for example. Or if you've been running a healthy donor with like 3-4 different panels over a year, you know exactly, and you can actually go back to the data that you've accumulated how things are supposed to look like. So for me, the quality criterion is how does my staining look like on that reference donor? And if I need to make adjustments, then my thinking is always, are those adjustments, do they have some rational cause? I know to use a common example, I know on that day I ran out of beads that I always use, and I used, let's say, a different type of beads that give me an issue with BV711, and now I'm seeing an issue with BV711 and per-CP Psi 5.5, which is exactly that same spectral neighborhood, and I know that those beads give me those issues. and I've seen it before how it's right and now it's not right, then I feel completely comfortable making an adjustment there because I'm having my reference control, I'm having my rational explanation of what went wrong, and I can adjust it. And then the third criterion that I have, and that I feel is the most important one, is there are certain markers that we stain where the biology is very clear. Let's say CD3 and CD19, or HLA-DR and CD123, where we know exactly what is mutually exclusive, how patterns are supposed to look like. And if I'm making any adjustment there, it's maybe primarily for visual pleasure, if you want to say so, or visual appearance, but it won't change anything about biology. While, for example, if I'm changing my unmixing or my compensation values, which in the end is the same thing, for expression of PD-1 and LAG3 relative to my CD3 on T cells, then I can actually change the readout drastically, change the readout of my assay, because I might introduce false negatives or other issues. And this is where, circling back to what Rui said in the beginning, where you might change the biological readout of your assay in a way that is not correct or does not correspond to reality. And that's where, for me, the cutoff would be. I don't want to make an adjustment that could in any way negatively impact the reliability of my data and the readout that I'm getting from it. That's how I'm thinking about it. But yeah.
Right. Well, and I agree completely the way you think about it and you're approaching it. I guess one of the questions I've had for a long time is what are the pressing reasons, right? Except for aesthetic reasons, and that's been one of the reasons put out there, but obviously that's not a scientific reason, right? But what are the reasons that lead people to need to adjust the data? I'll say it here.
I have a list of three. My list of three that I wrote for this presentation, like in this discussion, we have the question of, you're going to publish your data and nothing sticks out more than poorly computed or unmixed data. So you really want the data to look fine.
I wish every reviewer would see it that way, David, but it's not the reality, but I wish.
Fair enough. But in your heart, you feel it. And you
yes, I totally feel it, yes.
The second one, Rui mentioned it before, and I've heard it as well, but I have no idea what it actually means. But like whenever you're going to use high-end analysis, all these algorithms that try to look at the whole list mode file as a whole and try to pull these population apart, you really want your data to be properly corrected. I'm not quite sure what the outcomes are if it's not. So I haven't dabbled much with these type of data myself. And the last one has to do with this. This is a point we've made on this podcast. There's just no good controls, or no controls that will match your samples as closely as you would probably want. So there's probably the point we're making at the time is basically you're going to put a lot of effort trying to get the best possible controls you can get, but at some point there's a diminishing return in this effort, and you're trying to get insanely great control. You're basically, your PhD becomes not so much about fighting cancer, but at finding good controls for your mixing. Basically, there's an issue there. It goes back to the problem with the beads and the interaction with the beads and the fluorophore. If I listen to Kelly Lunson correctly, and I might not have understood what she's saying, but it sounds like she's saying like the way we treat the cells, the way the fixation, we're including a bunch of chemical reactions that will impact the signatures of these fluorophores all the time, and so there's just no way that your controls and your samples will be exactly the same, and so that's going to have an impact, and it's just unavoidable. And now the question is, okay, well, how do we move forward with this? And so these are the three main categories of justifications that I came up with. I'm not sure if I'm missing any. And of course, the cell sorting question is like
I mean, I was about to say,
people show up with poor data on the spot, and then you kind of go. But this one is, I feel like, is in terms of unmixing, you kind of go, well, just switch to traditional.
Right.
Don't try to and in my facility, we've manually compensated for quite a while using controls, and then if people don't have proper controls, then it's all. We tell people don't put our name at the bottom of the paper if you really want us to deal with this data. But
yeah, we haven't. I mean, let me since I asked the question, let me also unpack that a little bit. But I mean, I think except for the second case that you mentioned, where there is, you know, let's say the scientific reason to actually you know, have to adjust the data. I mean, the other reasons, you know, again, aesthetics or rush to publish a paper. I mean, there's no, right? That's not an argument, right? Oh, I need to publish a paper. My data doesn't look that good. We need to change it. I mean, if that's acceptable, then I mean, we're going to start doing that to everything else, right?
Yeah.
But so that is fraud. If you're adjusting the data because you need to publish that data, that is anyway. But I think, you know, again, the you know, coming back to you know, to these reasons, like the second reason that you mentioned, I mean, is it still considered enough justification to have to adjust your data, or you know, are we as scientists, should we go back and figure out what the problems are? And I think, again, Florian, you the way you address it by I mean, when you adjust the data, you're also adjusting the data based on a lot of references, right? A lot of other controls. And I should say also that if we think of it the other way, when I sort and people didn't bring controls and I didn't even compensate, you know, you saw where the population was, like a GFP population, you know, it's here, but you only have GFP. So I'm assuming this is GFP. I don't need to compensate, right? But on the other hand, it's almost like an adjustment, right? You're not also using the right controls. So you're not totally sure, or you may not be totally sure whether you're identifying the right population, right? So, so when I say that, you know, when I talk about data manipulation, you know, we could also argue that not compensating on or just leaving the data as it is could be also a way of adjustment, right? Of you're making assumptions of what you think the population is without controls, right? So so I'm just saying that, like, that's why I say we're not the gatekeepers of knowledge of the way of the perfect way of doing science. Nobody is, right? That's why I enjoy these debates. It's just trying to figure out better ways of doing things. And again, I think that adjusting the data after the fact based on your assumptions is a dangerous thing to do, because not everyone follows, for instance, Florian's you know, best practices, and we have a lot of I saw once an argument of, well, we're experts, so we know what we're doing, so we can adjust. The non-experts cannot, and that seemed very, again, we're going into, if we're applying non-scientific methods, the arguments are also going to be very non-scientific. So that's what we can expect.
I mean, one of the reasons why I saw people coming to me and without understanding what's going on, requesting adjustments or asking what's going on, and I think that's actually quite common, is that when they do their stats, not really knowing much about flow cytometry data, and they end up with negative values, which often come from overcompensation or over unmixing, if you want to call it that way. And there, I mean, again, if you have the right control, like an FMO control for that channel, it might be possible and suitable to adjust the data such that it's correct and actually better than it was in the first place. But that again circles back to the fact that you need to have the right controls. But since the question that you pose, David, is, or Rui, that you posed is, why are people doing it? I mean, I think often it's aesthetics, often it's because they're getting values that they don't understand, including negative values, which clearly shouldn't be there, or are an issue. And those are probably two of the main reasons.
Right.
Negative issue, you mean like overcorrection, or just the trumpet effect gives you?
No, because that shouldn't be, because the trumpet effect would, if it's properly compensated or unmixed, you should still end up with a median of zero, but not like only when it's like overcompensated or over unmixed, then you will end up with these negative values. And then people come and ask, at least I had that in a co-facility setting.
Right.
And the last reason, I mean, that goes back to kind of the non-spectral times, is really for all of you that are working with DIVA instruments, you know that DIVA wouldn't sort if the entire gate sits in the negative space. So in this case, yeah, you have to do it if you want to sort. So,
so. And you know, there have been, which I think is quite I don't know, for me it's motivating to see some companies, you know, not bring or avoid bringing tools that will facilitate this data adjustment. But on the other hand, actually these tools can be very good for troubleshooting, right? I mean, the times that I've gone to, I mean, I don't see so much and we can debate this, but the point of like when you publish sharing the unmixing matrix, etc. But or the compensation matrix, you know, what do the values mean exactly? But for, you know, practical purposes for troubleshooting, the times that we've gone there and we changed something and just, you know, this small adjustment, look at all the data. It's actually just a compensation issue or an unmixing issue, and that's help us troubleshoot and go back and try and fix what the problems are, right? So these tools are actually valuable, right? They're useful, but I do see some companies that, you know, decide, we're not going to put these tools out because this actually helps people. It's basically letting people know that, oh, if a vendor approves this, then it's okay, and so they want to steer away. And I do, you know, I see the value in that, but on the other hand, these tools are actually quite valuable, right, to be there.
I wanted to push back a bit on something Rui said, with the idea that if we need to spend more time on adjusting the data or the experiment in order to, before we publish, we should be doing that. But personally, my master's way back then was a terrible experience. And I developed a high sensitivity for my users, all these grad students. I just want them out of here so they can lead productive life. And I'm very concerned about anything that will delay that from happening. So whenever somebody comes to me with a question about their data, the unmixing is not great, what do we do? I really want to be able to provide better tools, better solutions than let's go back and do better controls, because like that's been the other bad answer I heard in a discussion we had at the ABRF was go do spectral mass cytometry, and
that's a very bad answer.
I blew a fuse there. I'm sorry for the.
No, but Dave, I mean, what you're saying, I think as a community right now, I'm of the opinion we need to be pragmatic. If there's data that has issues and we have a rational reason how we can fix it for that particular person, then I think it's okay to do this. However, we should avoid this as far as we can, work on the controls, and as a community, and I think that's the task moving forward, we really just need to come up with a freaking better way of getting things right in the first place.
Right.
So we need to have, you know, look, for example, like, I mean, I've been banging my head against the wall with like which bead works with which fluorochrome basically for like 7 years since I was working on the Symphony, and I had those ultracomp beads and the BV dies, and it just like was all a giant mess. And now in a way we still have the same. We have like 7 different types of beads, new fluorochromes, which might work with this bead, but not, I mean, not work, they will all work, but like which give a slight spectral mismatch on this bead, but not on that bead, and so on and so forth. And we should have like a public database, for example, that shows these issues, which as a community, we should be able to put that together. It's just no one has done it because you can't publish that high. And it's also hard to kind of make it in a visually easily accessible interface manner. And alternatively, and that's the bigger challenge, is we just need to come up with some particles that deliver the same spectral signature as cells all the time. And now we're in a podcast. I was saying this, why can't we just take some lyophilized cells and put that same capture element that is on those freaking beads onto lyophilized cells, and maybe that'll solve those issues. I don't know, but maybe a manufacturer will do it.
Right.
Yeah, anyhow, you know what I mean? So we need to kind of get now, so far we've been able to cut corners, like in the compensation world, it was so simple that we could do it. Now with unmixing, we might still be able to do it, but don't do it right. But in the end, we need to come up with a particle that allows the average user to just follow the pipetting scheme, do this, add the antibody, fix it the same way as your cells and run it, and it'll be right. That's what we need. And I think there is more investigation and community effort needed to make that happen.
Yeah, yeah. Sorry, I was just gonna, add a comment there. I think you're absolutely right. The community should be more focused on, rather than discussing whether we should be, we should manipulating the computer, manipulating them or adjusting the data or not. Rather than discussing that, we should be more discussing what are the tools that we can bring, that we can develop to make this easier, right? So that, as you were saying, David, so that you don't have to tell your researchers, oh, go back and figure out what the problems were with the single stain controls, which you might not even know and might not even be due to those single stain controls, right? So it is a difficult thing to do, and to ask a researcher to, spend most of their research trying to figure out the limitations of this technology. Right? It's people like us, right? We, the experts in full cytometry, should really be discussing, what tools can we bring? What better beads can we have, or carriers, right, for single stain controls? What are the issues with the instruments? I mean, manufacturers have told us that instruments are standardized for years and years, and I mean, even with spectral instruments, they're not standardized, right? There's a lot of
much more so than they used to be, though. One has to
admit. Yeah, and everything has been. I think I'm very happy. That's why, I thought it was very strange that Isaac would entertain the discussion of a society like Isaac should be actually pushing for these kind of solutions, rather than entertaining a discussion whether we should adjust data after the fact, after it's been measured or not. So I think, yeah, we should put a lot of effort in finding these kind of tools, metrics, increasing the level of standardization of the instruments, reducing the variability of the instruments, I should say, better fluorochromes, which we've seen a boom in the market with much better fluorochromes. I guess the beads are better than they were before, but we're also using way more colors together, so it also becomes more, any slight change will have an impact, right?
About the tools we're using, there were reports, many different reports, separate reports of beads being offering different signatures compared to whenever stained with cells. Sorry, fluorophores offering different signatures when they're attached to beads. The combination of beads and fluorophore is all over the place, and I've never heard any reports, and nobody came back and said, okay, we validated a specific combination that, or in other words, like I've people identified or confirmed that specific pairs of beads and fluorophore are actually reacting together, or is it just like
Dave? I mean, the problem here is we try to do is, because for OMIP 102, for example, we ran all the 50 single stains on beads and on cells, and we tried then to actually build the spectral signatures and see whether we can identify based on the spectral signatures where it works and where it doesn't. So where it works on beads as well as it does on cells. And the problem is that the mismatches in the spectral signature, in the normalized spectral signature, they are so small that you can't build a cutoff there. There are some like extreme examples, like for example the BB711. You can just overlay the normalized signature, and we have that figure in the supplement of OMIP 102, and for the record OMIP 69 had that already as well. And there you can see it because the spectral mismatch is large enough that you can just make a spectral, it's like a delta of the two normalized spectra, and you will see it. But then for others, the spectral mismatch is so small that the delta would be so small that you go like, that can't matter. But then if it's a bright signal on that fluorochrome, it's like it's a 0.5% error or 1% error, and still visually you would see it. And it's not only about the visual, of course, that would change the MFI of that secondary affected fluorochrome. So the problem here is really, even if you have that database, you can't easily like say, well, that's the measure or the metric where you can build the cutoff, because you really need to unmix it in the context of the full panel, unless you're just looking at those very extreme pairs like the BB711 and whatnot, where you can really.
Right.
So it's not trivial. That's why, we have that data. We didn't put it out there because it's like, I mean, first, no one would have probably cared, but anyhow, you get where I'm going. It's hard.
Yeah, we when we did this when we had this poster at CYTO comparing beads and cells, we were looking at the signatures between what we saw with beads and what we saw with cells, and even things like with similarity of 1.00 or 0.999999999, you still saw problems. They weren't the same, right? So there's more than just, or maybe these metrics are also not capturing how sensitive this is, but there's more, right, to it than just slight mismatches of signatures based on where they are, right? It could be chemical interactions with the buffer, right? pH. I don't know. There's so many things. Florian and I, we had a nice. Actually, am I still owing you a beer? I think I'm still owing you a beer.
Yes, you are.
We had a discussion during our spectral panel design workshop. What was the cause of? Was it BV785?
Yeah.
785 or 7?
Yeah, and the base, the BB421, the 785, and the base BB421.
And I mean, I still believe there's still influences from, I mean, neither carrier. We tested basically with the ultra comp, the spectra comp, and then in cells, and as you move from ultra comp to cells, it becomes more similar to the spectra. So definitely the carrier has a huge influence, but there are other influences, right?
Yeah.
That are also, and I think that's the difficulty. Maybe sometimes those influences are more on the side of the carrier, less on the side of other external factors, right? Like light exposure or temperature changes. But I think or pH changes, things like that. So I think we're just scratching the surface, and people like, I mean, I would say people like Florian, people like Oliver Burton, that are thinking about these issues and are trying to develop tools and ways to identify where are the problems, so that we can do better science and not have to adjust the data after the fact.
Yeah.
So I think
that's what is needed, and I think this should also be valued if someone does this. I mean, I've had discussions with Oliver. He's doing a lot of work in that space, but I mean, no, in the end, the currency of scientists is papers, and I think what we should do is get all 250 fluorochromes and run them on all 5 different types of beads. But who's going to pay for that, and who's going to give you a paper out of that? Right now, not too many people, so no PhD student is going to do it. And I would love to do it, but I have like 30 users sitting in the back of the core waiting to get their cells sorted, so I can't run like 1,000 samples just for the fun of it. Yeah.
Right. It's expensive though, right?
I mean, you need to get all the antibodies. We have like a large bunch of them, but you basically need to get all the CD4s and all the fluors and all the beads, and then you'll have, it's like 1,000 samples, and you might want to do that with like 3 different buffers. So we're talking about 3,000 samples. Okay. And then you need to be able to analyze that. And here comes the problem. That's actually where I gave up on it, because if you only need to analyze the normalized spectral signatures, fine. But if you need to start unmixing all kinds of different combinations there and manually look at where do you see issues, then this is like a year-long project.
Mm-hmm. Okay.
If you just want to do the math with the normalized spectral signatures, okay, but that won't tell you where, unfortunately.
Fair.
Yeah.
I agree.
Yeah.
Go ahead.
No,
that's a depressing statement now. So I'm just saying we can fix it. It just requires significant effort.
Just to put a like a ribbon on this one? Like whenever you do your research, is there one beads you use cells as much as you can, and then like move to beads whenever you have to? What's your or do you characterize every single combination before you run your panel and then?
I mean, this is kind of an independent podcast, so I don't want to put vendor names out there.
Oh no,
but
go
like
for it.
that...
Like this is the point of having an independent podcast, is like nobody's gonna yell at us. So go ahead.
Yeah, that's good. I mean, definitely, it's kind of the slingshot particles have gotten better, like compared to what we had available in the past. So I'm often using these now. Having said that, they also don't work for all fluorochromes.
Right.
So I prefer to use beads just for the ease of use, but in the past, I've only used beads. And then when we started doing OMIP 102, I moved gradually to only doing cells because we just needed to get like the most solid data. And then we always did both together. I mean, that was Andrew primarily. And now I'm usually doing beads for the floors where I know it works, and then like additionally cells for the ones where I know that there are typically issues. And then I settle on the one. But this is a lot of work, and I can do it first because I enjoy it. And second, as you stated, my goal is rather the tech development. But if you're a grad student that just want to measure those freaking cancer cells and you don't want to waste your mind on figuring out what's the perfect single stain control, which I completely understand, that's kind of our job. And this is why I would love to have a community resource that at least says, if that's the fluorochrome list you're using, here use beads, here use cells, and then you'll be fine.
Right.
Yeah, the thing is, it depends on other things, right? And you remember we discussed about the fact that there was once so we did several, during several days we measured beads and cells. So basically staining the same fluorochromes, the same panel, in different days. And what we saw is that when we overlaid all the or calculated the similarity index of all of these signatures, we saw that, for instance, in one of the cases, it was with Alexa Fluor 700, that all of the signatures were the same throughout all of the days, beads and cells, except for one of the signature, which was from the cells. So there might be also changes that are occurring within the cells, right? And in this case, we didn't even know if, okay, there was a change in the cell. So if we had stained another unstained sample, would it have been different also, right? In this case, would have been a nice test. But the problem is that it for us, at least in our hands, we don't have as much experience as Florian to systematically identify, oh, these always work, but these don't work that well. We saw a bit of a randomness there, which kind of concerned us because, best practices out there is that, well, test beads, whatever works, stick to that. Whatever doesn't work, use cells. But, in different days, that may actually change, right?
Yeah, but I mean, you're right. I mean, that's kind of an added complication. I mean, one prime example would be the PE Fire 810, because that's been in the literature as well. The PE Fire 810 is perfectly fine if you stain a marker that is on T cells, but if you stain a marker that is on myeloid cells, the myeloid cells that actually catalyze or seem to catalyze the breakdown of that particular tandem. So you are only going to see issues if you had stained something that is on myeloid cells and not on T cells. So yes, there is added complication to it, and like cells might change for reasons that we don't fully understand yet at all. But if we can get at least the basic stuff sorted out, I think that would be helpful to the community.
What do you think of starting with a smaller subset, and maybe this be a community effort? I mean, every time there are more people involved, obviously, then you need to standardize the protocols and make sure that they're all the same. But yeah, maybe we can do something. Maybe
let's keep that in mind for the next spectral course in January here in Zurich. So if we have some spare time on the analyzers.
Right.
How are these information currently shared? It's the first time I've heard of this issue with the Fire 810. So typically it's just like people use it and they like share emails or like
No, I mean, this is they mentioned it in the updated version of OMIP. So I've seen it, and a lot of other people have seen it in the updated version of OMIP 69. They were mentioning it,
Okay.
and Oliver Burton did actually some experiments, and I had some discussions with him. So I guess these days, whenever you want to find something unheard of, cool stuff, you need to look at the Colibri Cytometry blog. So just giving a shout out here. I agree with you. There is like no good way of sharing these small bits and pieces, but I guess that tells you something about the state of science. That when it comes to the nitty-gritty details of protocols, I think we are not doing a good job at sharing these effectively also, because even if we were to build, let's say, a community resource, how can we make sure that it reaches as many people as possible? That's kind of the hard part.
Yeah. As far as we're giving shout outs, I'm going to say Discord is a pretty good place to discuss with. Flow Stomping Discord is pretty awesome. Can we switch gears and talk about the manufacturers of instruments and their black box-ish unmixing algorithms? It seems to me that we had a full podcast on the SONY platform, and where we try to look at their like marketing claims and try to explain, because they kind of go against the fundamentals of what we've learned. But like it seems that SONY will use beads on everything all the time, and it doesn't seem like it.
Yeah, and it seems to be working.
Yeah, it seems to be working, and you go, and so there you have to assume that something about their unmixing algorithm is different, and we don't know what it is, because
it is for sure. Right.
Yeah, I mean, the black box thing, I guess that depends a little bit from which level you're looking at, because I'm sure there are a lot of people for whom compensation was a black box as well. So I think you can't necessarily only blame the manufacturers. Having said that, I agree. It would be nice that at least for those that are more into math and are able to kind of cook up things themselves, if it were all public information. But we are actually moving away from that, because every vendor is now coming up with their own proprietary improved version of unmixing. Having said that, I don't think that this will either introduce a lot of issues or solve the entire problem, because it's like always, when you have issues, you typically run the single stains and the samples on the same instrument. And my assumption is, but I might be wrong there, is that the algorithm won't introduce additional errors, if you want to say, but it's still kind of the same thing. But I might be wrong there. So
yeah, if it's a black box, we don't know, right? And that's, I think, as scientists, we never like black boxes, right? Because you don't know exactly what it's doing, and what kind of corrections it's doing, and what kind of pre-processing, post-processing is doing. So that's always a bit uncomfortable. But on the other hand, I mean, we're part of a scientific community. It's not the responsibility of the vendor to bring the right or the best unmixing algorithm, right? We can unmix after the fact, so we can use our own, develop our
just use like OLS,
right, the scientific community, and figure things out like that, right? If you don't like that black box that's showing you on the screen of the workstation, well, use something that right? Of course, that's difficult. Having said that, right, you need to understand how to use these tools, and the majority of the community doesn't. So they'll prefer to just use the black box, but that obviously makes people uncomfortable. So I think it would be more of the debate here, whether they should be more transparent in their in their tools. But I think it's not only in flow cytometry that we have these issues, right? We have it in every aspect of science nowadays. Actually, my first mentor, my background is a biochemist, and he used to, an old professor, he used to go crazy with, now vendors, sell kits. I mean, you don't even know what's in the kit, what the reagent A, reagent B. And I mean, you got a point there. It's a black box. We don't know which reagent.
Phenol extraction for RNA. That's the way to go.
Exactly. So I mean, but on the other hand, these tools are helping, and if they're validated by the scientific community, and like you said, Florian, right? You don't only compare you're not only using that technique, right? You're also comparing it to other techniques, to other references, other controls, your own data, published data. I mean, you're constantly comparing these things to build confidence in what you're doing, right?
I mean, the nice thing is the raw data is always there, as you say, Rory, so
Exactly.
we can always go and do OLS.
Right.
And I guess that's one of the good things, that every spectral cytometer still delivers the raw data as it is. And so everyone still has a chance to kind of work off that raw data
Exactly.
if she or he likes to.
Right. Now the thing is, sometimes some vendors implement what we perceive as better solutions, right? Like you were talking about SONY, beads, doesn't matter if it's bead or cells. I mean, it's not necessarily so, right? If the differences are enough, I mean, no algorithm is going to hopefully not fix that, because then there's some kind of manipulation there. But for instance, it would be nice to use that algorithm as a third party, aside, so that we could actually use WLSM or whatever method they're using, if their method is the best. But unfortunately, since these are black boxes and are protected, are IP protected, so we don't know exactly how to implement them, right? But yeah, I would say that that could have implications in which instrument you buy, because you may want to buy an instrument that where the data always looks better. But yeah, I don't know. I don't have a, it's not like it's a bad thing that vendors are generating good tools. What I think is, if it's a black box, then we as a community cannot say much about just whether it works or not, right? Better or not. But
I guess overall, I was just wondering if it's if we can expect the development of better unmixing tools to basically palliate the issues we have with the variation with our controls, or if that's a pipe dream.
I could imagine that there is eventually some approximation function or some AI guided spectral signature matching. I don't know.
Right. AI.
Well, I mean, so I don't know. I'm just gonna, and we're gonna be public, so I don't, I'm gonna try to explain this in the way I understand it. I have no idea if it's right or wrong, but the way I see it with the WLSM that SONY is using, you're basically providing an extra input of basically a vector of weights, and you can change those weights in order to fit better, as long as you have which usually there is a way to have a score of how good your fitting method is. If the fitting is better with changing these weights and fitting these weights, but the implication is that you're changing slightly let's say the signature, the input, by applying different weights to each channel. And by doing that, you get a better fitting. But those changes are going to be very, very slight, right? They're going to, and so any mismatch might still be workable, and provide you a good fitting, right? And I think that's probably why you don't need to have a sample that's as bright or brighter. In the case of this unmixing that SONY is using, which I don't know if it's just a pure WLSM or not, right? But that might be also, when there's a slight mismatch from a single stain control and the sample, it might not be enough to derail the algorithm, right? Like it would happen with an OLS without those weighted that vector of weights. So this is the way I see it. I don't know if this is entirely true or not, but it's somehow, right, it provides you a better unmixing result. May not always be better, right? It might just give you the same result because the fitting is what it is. But in general, it can. But the thing is, so why don't we implement WLSM in Flojo has that. So we can also implement and compare and see, if things are the same or not. So I think, again, it is a black box, but we can get around that. And as a community, we should be developing our own tools rather than blame the vendors for not having, not disclosing. I mean, as soon as the vendor discloses, all the other vendors will will do the same, right?
Right.
I don't know. I get it.
It's not so much on blaming the vendors as much as we've basically, when it comes to spectral, I feel we've now passed the puck of training our users to vendors. There's very little that's prepared by or made available outside of the CYTO meetings, outside of the like if a user who's basically naive in the world of flow stometry, just one of my grad students, for example, they don't know ISAC, they will not attend the 3-day training at the annual class or whatever. And so the main place they're going to get their information is going to be vendors at this point. And there's not going to be a whole lot of the training from vendors is often self-serving, or I should say, or
but also partially it's pretty good, I have to say. Some of them deserve a lot of credit for actually having pretty good learning hubs or learning centers.
But they're going to focus on the stuff that amplify their own technologies and so on. So you kind of want to be careful about.
Yeah, again, I think it's our responsibility. I've been very vocal in that. I agree, Florian, right? There are vendors out there that have done, an incredible job in putting out information, educational resources. And it's not just one vendor. There are many vendors that have really good educational resources. But as a scientific community, we need to be careful not to allow vendors to drive the narrative. That's the only thing I think is important, because, like you said, there's a huge conflict of interest, right? It always, you can even be driving in the right direction, but at some point, a decision may not drive you in the right direction financially, right? And then you start seeing things not said or things not completely transparent because of conflict of interest. And we don't know if that's happening or not, right? So again, just the same way we have to disclose all our conflicts of interest when we publish stuff. I mean, it's obvious when the vendor is providing training and education, we should take it with a grain of salt, right? Still critically read, listen to that educational content, making sure that you always test whatever they're saying, or you try to validate with your colleagues, with the community, and not just accept it as truth, right? That's my only comment there.
I'm going to step back to the new algorithms coming out, and Dave Novo had a pretty nice presentation at
that was pretty exciting.
I'm not sure it's going to impact the actual correction of the. We'll see when his paper comes out, but the actual correction of the spectral overlap. But the main point he presented is that whenever you have a really complex panel, at some point your target population only has a fraction of the total number of flow force that you have involved, and then that the unmixing applied to this particular subset doesn't.
I think it's not about subsets. It's really just a cell-specific unmixing. So basically, he developed a method where you can use only the fluorochrome signatures in that unmixing matrix of that particular cell that are actually present on that cell. So it's probably has to be some kind of approximation function or something like that. But it looked pretty cool. It's the same as, I think it looked pretty much the same as what the team from Greg Finnick at OZ presented with kind of they called it OZ Resolve, which I think conceptually sounded pretty similar.
Just to clarify, to our listeners who might not get it, but the general idea is like if you have 5 colors in your panels and you're unmixing, you set like 25 of these controls. Essentially, your excess variance or the error spread due to spectral overlap will increase because the model doesn't fit your data as much. So you really want the same number, same flow force in your unmixing strategy. So.
Right.
Yeah, I'm curious about that. I mean, he says the paper is under review. I mean, look pretty impressive, but it's kind of the same direction. No, you kind of adjust the unmixing to make it work better.
Right.
I mean, that's what he's trying to do. And maybe eventually there will be ways how we can extend that to account for like slight mismatches in spectral signatures between single stains and actual sample. So maybe it'll go that way in the future if we are lucky.
Yeah. Yeah, that's a smart way to do, since the more fluorochromes you use, the more chances you have unmixing dependent spreading.
Yeah, exactly.
So if you have a 20, so I think that the issue is more, you have a 25 color panel, but most of your cells will not have those 25 colors in them, right? Those 25 fluors. So unmixing as each cell goes by, right? And let's say they only have 8 colors, unmixing just with those 8 colors rather than the whole 25 could provide a better
resolution,
right? Better resolution, or at least reduce that unmixing dependent spreading, right, as much as possible. Yeah, but you see, these are, again, and I would say that David Novo comes from an approach of an academic approach, not as a vendor, right? And this is what we need. I mean, we need the community to produce better tools, and one day we won't even be discussing whether actually the vendors will be adopting maybe the tools that come from the scientific community. Who knows?
Right. So to wrap up, what tips do we give our listeners? Any grad students that is thinking of Florian discuss generating controls, actual model controls? That would be the main thing I'd take out of this discussion. Anything else we?
I mean, I think that's really what matters. What matters, not care too much about your sample, but care more about your single stain controls.
Right.
Yeah, and just critically, think about everything, right? Everything you're doing. I know that, again, because of the pressure that we have to publish nowadays, just doesn't allow us much time to think about all these complex technologies that we use. But well, think critically. And listen to your core staff.
Listen to the core director.
Right. Exactly. Listen to the core.
And any tools for these core directors so that they don't have to like send their user back to go do mass cytometry? Any extra tools we can?
I mean, there aren't. That's why we're debating this, right? There aren't systematic tools as of yet, right, that we can pinpoint exactly where. But I mean, there's already a lot of tools. There are already a lot of tools out there that help us with panel design. So what I would say is, attend expert meetings, right?
Training classes.
Right? Classes, training, educational resources that are out there. I mean, take advantage of all this and just learn as much as possible of what you're doing. Because
and here, if I may just add, people then always respond they don't have time for that. But in my experience, spending 3 hours in one of those classes will maybe save you like 20 hours of wasted experiments. So overall, it still pays off.
It does pay off. Yeah.
I really want those classes to be online. It bugs me to have to like travel nowadays. Like there's no money anymore, so we should do more online stuff.
Yeah.
I mean, there's the open flow stuff, which maybe doesn't touch on these topics yet, but in general, I think there's more and more good educational material out there. Some of which behind a paywall, unfortunately, some of which don't, and I think that's where we, again, as a community, can try to work. If we have some spare time, the three of us, maybe we can. Come up with
absolutely.
Yeah, if we have spare time, definitely
spare time.
Where is that?
Exactly. I don't know. All right. Yeah, you're right. You're right. And actually, I should say that we are thinking of bringing back open flow. That's been stagnant for a little because of that. A lot of people have given us really great feedback on your tools. Actually, there was even an emerging leader that was saying that they were inspired to follow up with flow and get into flow because of these open flow webinars, which is really, it's great to hear that the community is using, and a lot of people are in countries or in places where it's very difficult to get expert opinions to discuss with and go to these meetings.
Right.
So these online resources are really important. I agree.
Yeah.
And driven by the scientists, right? Not only by the vendors.
On the question of paywall, I'll say that so there's been discussions of setting CyTO outside of its little paywall box and back on YouTube, but I don't think it's going to pan out. But I'll point out that our website, that the CAD facility, is monetized and is actually making money, and so we use that money in weird ways. We just bought the Cytometry, so in order to like salvage it and save it for posterity. I don't know. There's many different models out there that we can share information that does not include that it's actually really cheap, really easy to do, and does not require a whole lot of investment. So there you go.
Great.
I'm gonna plug stuff. GLIIFCA is in Chicago this year. Registration is open. It's gonna be awesome. It's to do 2-day meetings, September 19th and 20th. Florian and Rui, do we have anything you want to
Rui and me, we are going to do the European version of the spectral panel design workshop, hands-on from start to finish, planning a panel, pipetting it, analyzing it. Rui will be there, I will be there, Sophie van Gassen will be there. So we'll announce date and schedule soon. So if you're up for that, it'll be in Zurich.
Yeah, the one in New York seemed awesome. So
yeah,
sorry I missed it.
This is exciting. I think again, a lot of people in Europe were commenting, "Oh, is there something like this going to happen in Europe?" And there we are. There we go. It's going to be exciting.
Sounds good. All right, thank you everybody for listening. Comment and like, and I'll do all the YouTube things, and we will talk to you later.